and the Compost Council of Canada to provide free learning sessions with incredible research and knowledge base who we might not be able to share at one of our events. And so we're very fortunate today to have the generous participation of Karen Bartlett, who is with the School of Population and Public Health in the Department of Civil Engineering at the University of British Columbia. And Karen has been generous in terms of doing this presentation at our regional workshop, Compost Matters in British Columbia, in the winter. And the importance of this information is it's so relevant to all our compost facilities. So we're very thrilled that Karen would be available to talk about her subject matter and the research she's done. Karen is a professor in the School of Population and Public Health. She holds a master's degree in Occupational Hygiene and a doctorate in Interdisciplinary Studies from the UBC. She received post-doctoral training in inhalation toxicology at the University of Iowa School of Public Health. Dr. Bartlett's research interests includes indoor air quality and bioaerosol exposures in work and community environments, particularly in rural and agricultural settings. Her degrees and her recognition amongst her peers are tremendous. And so without further ado, I'm going to turn it over to Jolene and Karen. And on behalf of the Compost Council Canada, and very importantly, Green Manitoba. Karen, thank you very much for sharing your data and information with us.
Well, thank you, and I am absolutely delighted to have been asked. So I'm hoping that people will find this interesting. So what I'm going to be talking about today, just make sure that my slides advance. Okay, now what's wrong? Oh no, why aren't my slides advancing? There we go. Okay. We're all good now. The compost, everyone on the line, I'm sure, is familiar with what is compost, and so you're wondering why I have this slide up. But part of the actual process, integral to this process, is part of why I need to talk about exposures to the compost workers. And so, as you all know, it's a microbial process that breaks down the complex structures of the feedstock, and in that process generates heat. There's some, the graph is of actual data loggers that we had implanted in the piles, in windrow piles, to document that heat regimen. Now, we have a number of different feedstocks that can be used, that can be composted, obviously yard waste, food scraps, and biosolids, and that is, I will mainly be talking about food, yard waste, and biosolids today. So in that process of the increase in the heat, it kills the pathogenic bacteria, which is what we want it to do, so that we have a product in the end that is safe to use. But at the same time, what it's doing is encouraging the growth of thermophilic organisms, both fungal and bacterial. So it's those thermophilic organisms, or the heat-loving organisms, that I'm going to be focusing my talk on, because as it happens, some of the components of those organisms can cause problems, particularly when inhaled for workers. And the reason for that, you'll see that I have an illustration of an Aspergillus structure seen under the microscope. And that structure has a very small spore. I've got pointing to it about 3 micron. And those spores are released by the millions upon millions when the organism is disturbed. So when we look at the human physiology of the worker, we have I have a illustration here of a person, and nasal area going down into what we call the thoracic area, and eventually into the lungs and into these little tiny organisms, organs, which are called the alveolar sacs. That's where our air exchange happens. So we have workers, more and more workers are coming into the workplace. The scale of the municipal composting is becoming larger and larger. And so we need to start paying attention as we bring these new facilities online to the health and safety of the workers. And part of the challenge is that we've kind of self-regulated up to this point, where if a person is having trouble, they leave the profession. And if we can plan in advance, we can actually make it a safe place for people to work as we expand our facilities. So as air is inhaled through the nose or the mouth, the dust or the particulate matter, or those little tiny spores that I talked about a minute ago, will eventually end up somewhere in the respiratory tract. And in addition to the workers being exposed, as we have this rural versus urban divide is becoming less of a divide, particularly here in British Columbia. So we need to also think about the members of the public who are coming to facilities to offload feedstock and things like that. So when we're looking at those little spores, where they end up in the lung is way down here where the alveolar sacs are, and that's why they can cause a problem for people and for workers. So we did a study because we specifically wanted to look at what the exposures would be to then airborne dust, microbes, and microbial products that are generated in the composting processes. We also wanted to determine which tasks that are unique to different feedstocks and or facilities would be potential for higher exposures, and to evaluate control measures and also that aspect that I mentioned before of community exposure. So this illustration is our student who is affixing a monitoring device to a front-end loader, and that was one of the examples of some of the samples we took. So we did most of our work in 3 different sites, and we wanted to look at a variety of climates and a variety of feedstocks and or processes. So our first site was located in a high desert kind of climate, so hot, dry summers and very cold, dry winters. In this particular case, their only feedstock was biosolids, and they were processed with static aerated piles. And here we have an illustration of the blending of the biosolids with bulking agent within a tent-like area that we're going to be talking about in a few minutes. Our second site was also located in a high desert, cold winters, warm, dry summers, and they had part of their facility processed yard waste, the other part biosolids, and their process was windrow piles, and they had a high, this is building the pile, and they turned the pile with this high energy turner, and we will look at that in more detail in a moment as well. Our third site was located coastal rainforest, so wet but mild winters and somewhat more wet summers. The only processing yard waste and the process was in windrow piles. You can see the building process here. So we looked for different components, and I wanted to just spend a minute or two talking about why we looked at the components we did. So dust, or particulate matter, that's what the PM stands for. Any particulate matter that's less than about 100 micron in aerodynamic diameter can be inhaled. Now, only the very small particulate will end up in the deep lung. But we also have lots of receptors in our upper respiratory tract that can react to what we call allergens, and or allergens are very readily part of what we call organic dust. So that kind of dust that we see in the rural industries and things like that. So part of in addition to the allergenic component of the organic dust, there's a component called endotoxin. And endotoxin is simply a biochemical. I've given you a structure there that's associated with gram-negative bacteria. Now, our world is full of gram-negative bacteria. Probably we would find more in bio-waste, but they're really gram-negative organisms are everywhere. And as part of their cell membrane is a component that acts as what we call a pro-inflammatory agent, so that when it's inhaled into the lung, it sets up an inflammatory response in the lung. So we can take the dust and look for this particular active ingredient, so to speak, of the dust. And that was part of the chemical analysis that we did. In addition, we took shorter-term samples of two viable marker organisms, one called Aspergillus fumigatus, which is a fungus and makes those very small spores. In some people, it can also be a pathogen, depending on the person's age and or immune status. And another group of organisms that we grouped together and called Thermoactinomycetes. They're bacteria. They also form spores. And we know from research that the combination of Aspergillus fumigatus and Thermoactinomycetes and endotoxin, that combination for some people can be a real problem. Whoops, didn't mean to do that. Can be a problem in with a disease called hypersensitivity pneumonitis. It's an allergic disease of the lung, and people who have this disease typically will have to leave the industry. In fact, in some severe cases, have to move to an urban environment because of the reactivation of that particular disease. So let's go to the results. So remember, we had those 3 sites, and our first set of results is a comparison of the 3 sites across this way, and a comparison of inside cab concentration, outside cab. That was what that illustration of the student placing the monitor was, outside cab. And then for 2 of our sites, we also had personal samples, and that was when we put the monitor on the worker, him or herself, and was able to compare the general outdoor environment to the actual worker exposure. So you'll see that site 2, that high desert with a high energy turner, had significantly higher exposure in general to the dust, but also the personal exposure was quite high at that particular site. Now, when we look across the sites and compare the outside exposure to the inside cab exposure, this is the good news story. Not only highly significantly different exposures, but we can definitely make the recommendation that if the cab in the equipment, front-end loaders and such, are properly maintained, that we've actually reduced the amount of exposure. And as you can see from site 1, that personal exposure was almost just as low as the inside the cab exposure. Now, what happened in site 2 was that the very hot environment in the summertime, and the air conditioning wasn't working as well on that particular high energy turner, and so the worker had the window rolled down, basically. So we know that not only if the filter is being maintained, that we get a good reduction, but we have to also maintain ways of keeping the worker comfortable in that environment. In this particular case, the air conditioning has to work. And that's just a picture of the high-end turner. So you can see the enormous amount of dust that's being created there. And when the window is rolled down, that dust is being able to enter the cab. So now that specific chemical that I talked about, endotoxin, in this particular case, Site 1 now turns out to be the highest exposure. And again, though, the outside exposure was considerably higher than the in-cab exposure. So we know that we're getting protection. And the reason for the high, just go back to this for a minute. And in all cases, the cab was able to protect against exposure to endotoxin. Again, we have a remnant of the problem with having the window rolled down in Site 2. Now, endotoxin is not currently regulated in Canada. In Europe, it is as an occupational exposure, and that level that is used in European Union is around 90 endotoxin units per meter cubed. So you can see that with proper maintenance of the in-cab, we actually can achieve that occupational exposure reduction in the actual exposure. Now, the reason that Site 1 had such a high outside the cab exposure was partly because of the actual feedstock itself. It was biosolid feedstock, but because it was being contained inside that tent. And when we presented our results to the actual facility, we made mention that if they could improve the ventilation in that tent, or at least align it so that you had wind blowing through it, you could reduce the amount of dust inside the tent. Our understanding was that they took us up on that, and that ventilation has been improved after our presentation to them. So now moving on to more short-term samples. These were taken for the Aspergillus fumigatus, which is the fungal component. And I want to point out that on the scale here, this is a logarithmic scale. So each one of these hash marks is tenfold greater than the other. So we have to take that into account when we see those that are high exposures are quite a bit higher than the lower exposures. Again, we have in orange Site 1, this olive Site 2, and gray Site 3. Now, here we've incorporated the task with the highest exposure here was a blending. And because blending only happened at Site 1, we can see again that not only was the endotoxin elevated, but that process was elevated for exposure to Aspergillus fumigatus for the same reason that we talked about a minute ago. Now here we're looking at the upwind-downwind exposures. So upwind is what would just normally be present in a rural environment, an agricultural environment, which many of these, our 3 sites, were still kind of considered agricultural. And so if we compare now, we want to compare the upwind to the downwind. What we're talking about downwind is what community exposure may be if the community starts to move closer to the facility. And so there is an increase overall in our all 3 sites. There is an increase of downwind exposure, but not as great as the task-based exposures. But it's something that we want to keep in mind because members of the public may not be as robust as workers are, and so we have to think about how we're going to protect them from some of these exposures as well. So thermoactinomycetes in general is a lower exposure. Again, we have a logarithmic scale and different tasks. And they were, some were screening, for example, in Site 3, resulted in the highest exposure for across the boards for thermoactinomycetes, and not too surprisingly, grinding and building the piles would be higher exposures And as it happened with what we call the low energy turner, it was the highest exposure in the set of exposure analyses. Again, we're looking at the upwind downwind exposures, and here we see that there is potential for some downwind exposure. Again, we're still diluting. There's a dilution effect for most of the downwind. So by the time you get to the property line of the facility, you are diluting these viable organisms. And we need to just remember that to keep it in mind when we're designing our facilities. Just going to go back for this for a minute. And the other message that when we took these, when we did this study, it was a 2-year study, and we incorporated into the means that I'm showing you today different seasonal variation as well. Now, if I were to do a sub-analysis, I could tell you that the summer exposures were quite a bit higher, simply because of the dryness of the process. The winter exposures tended to be very low, partly because some of these facilities, in fact, just overwintered, and there wasn't as much activity at the site over winter. And in the coastal rainforest, it just got washed out of the air. So rain can wash these exposures out of the air, both the particulate matter, the organic dust endotoxin component, and also the organisms as well. Now, I wanted to take this chance then to also move on to a newer study that we're just starting to work with now. And you remember that when you enclose a process, we tend to get higher values on some of the measures. And so we went to look at a transfer station. And so you probably have seen these. We've got a crane that is moving these materials down below. We've got a front end loader that is loading the materials into transport trucks. The transport trucks then go out to the site. Now this is a combination, of course, you'll see, of municipal garbage. I don't have it in the picture, but as well off to the side, members of the public could also bring their yard waste and compostable materials. And members of the public, again, could drive up to the edge of the pit and dump their own materials into this facility. So this was what it looked like. It is enclosed, and these are some of the results that we ended up with. So again, now we're looking at endotoxin, and we look at the sampling location was the pit floor, and we were put a sampler on the worker, and that shift long concentration was 933 endotoxin units per cubic meter. And the safety watch station, we put that in just at the safety watch area. People aren't in that area in the same intensity as on the floor pit, and so that is reflected that the concentration was a bit less. Inside the dozer versus outside, so this is outside, inside, and inside and outside crane, outside and inside crane. So the good news story, again, is that when the dozer is in operation and the filter and the air conditioning and to be able to make it comfortable for the worker is in place, we actually are holding the exposure less than the European Union suggested 90 endotoxin units per meter cubed. Now, what it means, though, when we look at all of these data together, is that there was a worker who was exposed or completely overexposed to endotoxin. And it's that constant inflammation that we think can cause problems. Now, for this particular worker, could either wear a respirator, and in this particular case did wear a respirator. So this is a reason that we would want to go back and say, okay, we know that the respirator is uncomfortable, but until we can put in better controls for you, you're going to need to wear the respirator, and this is why. It's because the endotoxin concentration would be high enough in your area that we think would be hazardous to your health over the long run. But the good news is that those folks that are working in the different containment areas on the floor don't have to wear the respirators because, in fact, we've been able to control that exposure. Now, where this could be the good news story going forward is if this facility were to be redesigned, and we have it on good authority that there probably will be a redesign at some point, is that we could look at, again, targeting specific kinds of ventilation within those facilities to be able to reduce these kinds of exposures. Now, we also looked at the thermoactinomycetes and the Aspergillus fumigatus like we did before. And you'll see that we're looking at colony-forming units per cubic meter. And so this is upwind, remember, is just where it's in the background. This is an area that's kind of at the edge of town, and so has lots of blackberry bushes and various and sundry things. And so there's going to be some circulating organisms in general. The downwind wasn't too bad. And when we say downwind, we're actually talking about at the property line. So this would be, for example, if you were standing at the fence line. Now, what is interesting, though, from these data is here is the public tipping floor, or if you were standing by the crane. So we do start to get a little bit worried when we get these high exposures that could potentially be experienced by the public, and for the employee that is just simply near the crane, or we were also looking at the truck drivers who were sometimes with their, waiting for their truck to be filled before they pulled away. So we start to look at some of these exposures, and we get a little bit worried that, again, we would want to maybe look at these data when the facility is redesigned, because we think that is the point at which we can actually affect a change. Now, these two samples were taken in the lobby, and typically we would call these office worker exposures. I think the lunchroom was in kind of this area as well. And so it's just a reminder to us that these very, very small particulate matter can be carried with air when you're opening and closing doors and that sort of thing into an interior space. And once the organisms are there, they tend to be able to survive. Both of these organisms, Thermoactinomycetes and Aspergillus fumigatus, can survive desiccation. So they can actually, be in an area that you weren't actually expecting them to be. So in this particular case, our recommendation was to put positive air pressure in the office lobby areas and to be able to look at opening doors that directly onto the tipping floor, that we could reduce those exposures as well. Now, these exposures, I wouldn't say are immediately hazardous, but remember that you've got employees that will be coming as we build more and more facilities. We will be bringing workers that may have pre-existing allergies, for example. Aspergillus fumigatus is one of the fungal antigens that's often used when you're diagnosing fungal allergies in asthma and things like that. And so it is a common antigen that people can become sensitized to. Now, if you bring a sensitized person into this kind of environment, these kinds of exposures could trigger an exacerbation of their condition. And what we want is for workers to be able to safely work in these environments and not have to leave the industry should they become sensitized because of their work in this environment. So in conclusion, and I'm leaving lots of time for questions, but take-home lesson, CAB offers a reduction in exposure both to the dust and to the bioaerosols, but it must be maintained. Filters must be changed regularly, and you have to provide for the worker to be able to survive in an enclosed space like that with air conditioning. Endotoxin, you're always going to have a higher endotoxin concentration with biosolids as your feedstock. So again, keep that in the back of your mind, and perhaps look at the ventilation conditions. Open air ventilation has quite a lot of dilution effect, and it's as soon as we put a containment over it that we start to build up concentration. So that's the point that we need to start looking at controlling that. And the task analysis is a good way of looking at the actual facility and to see whether there are additional controls that can be implemented there, and or if there are tasks that are performed by the worker that may need to be controlled with wearing a respirator. Even though that respirator I know is not a popular thing to wear, I totally agree, but now that we see what it is that is being controlled, we need to work out how to control that. So I whizzed through that. I've got my acknowledgements. We were funded by WorkSafe BC, and we had a whole wonderful crew of students that participated in our sites, and all of the workers at the sites were just absolutely fabulous. And so with that, I will answer any questions that you might have.
All right, thank you, Karen. This is Jolene Rutter from Green, Manitoba. I'll be moderating all your questions, so if you do have a question, currently there isn't any right now, but you can either type it in. There's a little chat dialog box. You can type it in and I'll read it aloud for you, or you can, there should be a little hand that you can click on, and then I will unmute your microphone so you can ask Karen yourself. So the first question is from Pat Miller, and he's asking, is there any type of respirator recommended for compost workers?
Well, the respirator would need to be a fitted half-face respirator with a P95 or a particulate arrestance filter would be the best, and but a half-face would be sufficient. But of course, the tricky bit is that to be fitted, there can't be any facial hair, which would interact with the seal of the respirator. So it's that's why kind of taking your respirator into the cab while you're in the cab, probably you're getting enough protection. But as some of my data showed, is that walking across the facility, for example, that those outside exposures can sometimes be higher than we would have expected them to be.
All right, and the next question is from Jackson, and he's asking, are there any safe levels for these microorganisms?
Well, that's a really good question, and I'm going to digress just a teensy bit so before I answer it. One of the challenges of why why we chose the agents that we did choose for this particular study is that Aspergillus, a fungal antigen, and or Thermoactinomycetes, which is a bacterial antigen, is that if a person becomes sensitized to the agent, then no, there is no safe concentration. And it's simply because our bodies have, for a sensitized person, our body will react to very, very low concentrations of that antigen. Now, for a person without a pre-existing allergy, and for a person who does not have a genetic predisposition to form antibodies, they can tolerate a higher concentration without any problem at all. And it's as though, you know, there's nothing, almost nothing in the air, you know, until you get into much higher concentrations. So, for example, if a person were to have pre-existing asthma, because some of these antigens are associated with the antigens that asthmatic people do become allergic to, it would be difficult for, unless you were to protect a worker, it would be difficult for a person with asthma to work in these kinds of environments without respiratory protection. Now, the CAB, the fact that we were able to decrease the exposures over tenfold in all cases in the air-conditioned cabs, when the cabs at least were properly running, would indicate that if a person, even if a person were tended to become allergic, they should be able to safely work in the cab without having to wear respiratory protection. And so that's why we say if we could also reduce the exposures in any other enclosed space, so for example, the tent, if we could reduce, and or the transfer station, if we could reduce those exposures as well, then we just have very much less chance of causing a problem. Now, and you saw that we were in fact able to reduce the exposures to below the European Union suggested guideline of 90 endotoxin units per meter cubed. And again, we think that that was based on a health-based standard, that people who were exposed to less than 90 endotoxin units didn't show any of the symptoms of inflammation in the lungs. So we can get there. And once we reduce those exposures, much less likely that a person would become sensitized and or have any long-term effects.
Thank you for that response. Very thorough. Touched on a lot of important topics. There isn't any new questions. Oh, okay. I always say that, and then one just always comes in. And this is from Shannon Ripley. Hi, Shannon. I work with Shannon quite often with the CCME. Her question is, how often would you recommend that the filters on a half mask be changed? I likely have been sensitized to Aspergillus and wear a P100 mask when at our local compost site, which I find has worked well, but it would be helpful to know how often it should be changed.
Well, that's an interesting question. And with the P100, P95, the P100 is slightly more, will block slightly more size relationship than the P95. So P100 is certainly a good one to go with. The way that respirator filters work is that all modern respirator filters, the P100, P95 series, are in addition to being a mesh of fibers, if you were to cut that thing open and look at it, it's a mesh of fibers, is that those fibers have an electrostatic charge on them. And so they are efficiently removing the particulate matter from the airstream, both by impaction and by actually making, attracting the particle, that little dust particle, to the fiber so that it can't make its way through that mesh. And so which is a little bit different than when we picture a filter just simply having holes in it and the dust particles kind of making it through the hole or not making it through the hole. So as the filter is used, it actually becomes more efficient for particulate matter because up until a certain point. But as that mesh attracts and doesn't release particulate matter, it actually becomes more and more efficient and can trap that particulate matter. Now, what happens though, and I can't give you off the top of my head what an exchange out schedule would be, but it would definitely be at that point when it's difficult to, when you're finding that it's more difficult to inhale through the filter. You'd want to change it out before you found it difficult to inhale. But they can, for the particulate arrestants, it can go a little bit longer than, for example, say chemical arrestants, because of the fact that the filter just keeps getting more and more efficient at removing the particulate matter. And I think another thing to remember about the particulate arrestants filters like the P100s is that they are rated for what we call the most penetrating particle size, which is 0.3 micron, which means that they are extremely efficient for the kinds of size particles that we talked about in being able to inhale or being generated by the dust in these kinds of environments. So the P100, P95 are terrific for this environment.
All right. Next question is, how is EU measured in terms of counts?
Okay, so endotoxin units are an actual functional relationship. And so we collect the dust, we bring it back to the lab, extract the dust out of our monitoring equipment, and we then expose a very, very sensitive biologic agent to that dust. And the biologic agent will go through its reaction when it encounters an endotoxin molecule. So because that biologic reaction of endotoxin plus the reagent in the system so closely mimics what actually happens in our body, it's what we call a functional assay. And so the European Union, in setting the limit at 90 endotoxin units per meter cubed, has a very robust, health-based way of determining that. So, for example, if I were to just weigh the dust, it would not be as good of a measure, because even though I could, make some guesses about how much endotoxin it actually contained, but by being able to go that extra step and actually look at the endotoxin units as a functional unit itself makes it a highly specific and a highly sensitive measure, particularly in these organic dust exposure kinds of environments, because it is so specific to that particular reaction that happens in the lung as well.
Very interesting. I hope all the scientists on the line enjoyed that as much as I did. The next question is going back to health. Are there other health effects other than allergies? Do other health effects occur other than allergies from these microbes?
When we look at particularly the combination of the microbes and the endotoxin, the lung has to deal with clearing the dust, or the dust, whether it's a particulate matter, what we call particulate matter, or whether it's the actual spore, is that we have in our upper respiratory tract, so the nasal cavity, we've got all those hairs right in our nose. And so a lot of dust is filtered out at that point. And so when you blow your nose, and right, we all know that. But the smaller particles can evade in the upper respiratory tract, can evade all those nose hairs and get down into the lung. So the lung has specific binding sites that will, when the binding site recognizes endotoxin, will cause inflammation in the lung. And so for people who are not already sensitized, there is a condition that's called organic dust toxic syndrome. And that's a whole long, huge word for being exposed to a short burst of very high biologic organic dust. And typically the symptoms of that are short-lived, but it's like having the flu. It's kind of running a fever, feeling chest tightness, feeling like you're coming down with something. And the good news is that it's self-limiting. It's just the way our body is dealing with that particular exposure and is clearing it. But part of that clearance is that it's set up a cascade of biochemicals in the lung, and that's what's making us feel like we're coming down with something, because it's a generic kind of reaction that happens. And part of that is we're trying to get that dust back out. The body is trying to get that dust back out of the lungs. Now that's a short term. So anybody, regardless of whether they have allergies or not, could potentially have that reaction. And in fact, I can testify that even I have had that reaction. And so that's how come I can explain it so succinctly, is that it's a short term, it's self-limiting, and it simply means that you walked into a situation or were at a situation where there was a lot of microbiologic and agents in the air. So if we were to take that situation now, though, and it wasn't just a one-time or unusual exposure, is that even if you don't have allergies to the antigens, the Aspergillus, for example, antigen, the body needing to deal with that mixed exposure can potentially result in a condition called hypersensitivity pneumonitis. The common word for that is farmer's lung. And not too surprising, it was named because we first recognized the disease in farmers who were being exposed to long-term exposures to organic dust. And what happens in that situation is that that inflammation, which is the body's way of dealing with the exposure, the inflammation becomes chronic inflammation. And at that point, it becomes more difficult to treat, and there are some longer-term health implications for that. So we want to stop the process. We want to stop the exposure before it gets anywhere near a chronic inflammation kind of scenario. But short-term exposures, our bodies are incredibly robust. And so for most normal people, it wouldn't be a problem as long as we can reduce those exposures. Now, whether a person with pre-existing asthma, for example, could ever work safely in these environments is another issue altogether. And so I won't go into that. So I'm really talking about healthy workers going in, but keeping them healthy for their working lives.
And on that note, Matt asks another question here. Has there been any health-related issues reported as a result of composting biosolids outdoors?
To the workers, yes. We have had WCB claims for hypersensitivity pneumonitis. Yeah. We have not had any complaints from members of the public.
And specifically with the composting biosolids outdoors, or was
oh, I'm sorry. So
indoors?
Specifically to biosolids, I would say that I think
it's specifically with outdoor. Yeah, biosolids and outdoors.
There have been claims. Wasn't specifically to biosolids. And it was basically, yes, but it was workers that were, you know, in the condition of, for example, turning a pile, not being in a cab. So it's an older technologies. And so we've not had any claims in people who have come through their working life being able to work in cabs and things like that. But it would tend to be a person who was really at the source rather than a person who was, for example, just walking across the facility or things like that.
So it's much more common in indoor facilities then?
Yes.
Correct?
Yeah.
Okay. And the last question here, hopefully, is from Michael Rourke from Ontario, and he's asking, to your knowledge, are there labs capable of testing endotoxins located throughout Canada?
That's a good question. I believe that there are. The samples can be sent to labs. There are several labs in Canada that can do it, and the samples themselves are pretty robust for being shipped around. So yes, there are. But off the top of my head, would not be able to give you the addresses. But yes, they are available. That test.
Okay. So lots of comments on thanking you very much for your information. Oh, there is one more here about what kind of precautions would someone with pre-existing allergies or mild asthma, what type of precautions would they have to take to safely work in a composting environment?
Well, my suggestion would be to, maybe that they would need to wear a fitted respirator. If they were working in, for example, the office environment, if that office environment could have a slightly pressurized relative to outdoor air handling system, so that just reduces the amount of dust that can get into the office space. Insist on working in a cab that has air conditioning and has well-functioning filtration system. I think that a person would be able to work under those conditions, and assuming, hopefully, that particular, asthma is a disease of specific antigens, and so if the asthma may not be brought on by the antigens at a compost facility if they weren't already pre-existing, and if we decrease the amount of exposure to it. So it is possible, but it would require, being in conditions where we know that we've reduced the exposure.
Okay, and I actually have one question. And that would be, so when you do these studies at these sites, are people surprised by the results, or is there some particular area of focus that that they didn't know?
They typically are surprised, and I think the reason is that we all know we all know that there's dust, right? Okay, so that's not the surprise, right? But it's digging down into, well, yeah, we know that there's dust, but what is actually in the dust? One of the situations that I learned about after we did this study is that sometimes, and this is actually going to become more of an issue, is that it's possible to use gray water to dampen down, to hold down dust, and gray water will have a higher endotoxin component to it than just city water or other kinds of water. So it's not to say that I have anything against using gray water to dampen down as a dust suppression mechanism, but just that we have to be aware that gray water will have some endotoxin component to it. And so again, better to be in a cab that is closed and with an air conditioning system, or if you were very near the source of the spray from a gray water system, to consider wearing a respirator.
All right. Certainly some things to think about when we're operating out in those composting facilities. So thank you again on behalf of Green Manitoba for the information you've provided here, Karen.
Okay, and I'm very happy. I'm hoping that somewhere there's my email address is going to be available to people. I'm very happy to answer questions, and feel free.
That's fantastic.
To do so.
And this presentation will be available on the Compost Council of Canada website. We've also
Terrific.
recorded the video of it, so presentation slides and the video will be available. Danielle, or
thank you so much, both Karen. The information is incredible, and I'm so glad that there was such a wide range of questions presented, because obviously a lot of people were getting great value out of this. And thank you also, Jolene and our friends at Green Manitoba for hosting the webinar. Thank you, everyone, for participating.
And thank you.